All right, so it is a pleasure for me to introduce today's speaker, Dr. Roundtable-Lasseigne. She is an assistant professor in the Department of Cell Development and Integrative Biology here at the University of Alabama at Birmingham Medical School. She is going to present on prioritizing polycystic kidney disease drug targets and repurposing candidates from pre-cystic and cystic mouse model gene expression profiles. So without delay, Brittany, I'm going to turn this over to you. Present as soon as you are ready.
Great, thank you. Let me get my screen set up. Can you see my slides okay?
Everything is good.
Excellent. Well, thank you so much for the opportunity to speak today about our work. I'm really excited to share with you what we've been working on and to hear from folks here in the RRC. A little bit of background about our lab. So we're a genomics and data science lab with the goal of discovering biological signatures to improve patient care and provide insight into molecular processes contributing to disease. We have two related research programs in the lab. So the first of these is investigating the impact of cell and tissue specific regulation and expression, cell-cell communication, and sex-associated molecular changes on disease manifestation. And the second is using computational approaches to identify optimal precision animal models and nominate and prioritize drug targets for treating patient cellular phenotypes. So since moving here to UAB, we've been researching PKD because we're really interested in the opportunity to try and understand cell and tissue specific context dependency and to try and look for drug opportunities. So while PKD is characterized by kidney cyst growth and volume expansion, there's a lot of phenotypic variability, and it is a multi-system disorder with extrarenal manifestations. And so we're interested in trying to understand the molecular underpinnings and identify drugs and drug repurposing targets. So in our work thus far in the lab has been focused on AD-PKD. So everything I'm going to be talking about today is centered around that. What I'm not going to tell you too much about today is some of our work in identifying disease mechanisms driving cyst initiation or disease progression. And that's been work that's been closely here with the Yoder, Mrugh, and George labs. But as I mentioned before, just in brief, PKD1, not only is this a multi-system disease, but PKD1, PC1, and PKD2, PC2 are expressed in several different organ systems and many cell types. So for example, here on the left, you can see the expression of those genes and protein products across different organ systems in the human body. And this is in healthy individuals. And on the left, I've shown you just one figure of some work that we've been doing trying to understand the effects of PKD1 knockout in silico across different cell types. And as you can see here, we find that proximal tubules are predicted to show enrichment of known PKD1 targets and metabolic expression changes, while macrophages demonstrate changing hedgehog signaling, et cetera. So this further just underscores how cell type specificity plays a critical role. So hopefully I can come back and talk to you guys about this work a little bit in the future and about our work in cell-cell communication. We're also working on long read sequencing approaches, which I think is going to be really important here in PKD, where we're trying to understand differential transcript usage and the effect of different variants. But like I said, I'm not going to talk about that work today, but I'd love to come back and share in the future. What I am going to talk about today is really talk about drug target candidate identification via computational biology approaches and how we can prioritize those different targets and candidates via cross-species considerations and by trying to manage predicted adverse events or side effects. So as this room surely knows already, tolvaptan was approved by the FDA in 2018 as the first and only AD-PKD therapy for adults with a high risk of renal cystic disease progression. So it's been shown experimentally to suppress ERK-mediated cell growth, fluid transport, cyst expansion, and reduces the rate of renal functional decline by 30%. However, it doesn't cure AD-PKD. It doesn't show additional benefits to other disease manifestations. It's associated with liver toxicity, and at least today, it's very expensive. So we want to work on trying to find other biomarkers of disease progression or new treatments for these patients. All right, so it's important for thinking about targets. So we know that AD-PKD is most often caused by mutations in PKD1 and 2. And here, the work that I'm going to share with you today largely centers around data reanalysis and PKD2 models. So for the majority of my time today, I'm going to walk you through some work that was led by Lizzie Wilk, a really talented computational biologist in the lab, and then try and save the last 5 to 10 minutes to kind of talk about where we're going with this. So this work has been pre-printed and it's in revision. So it's on bioRxiv. You can go check it out if you like. And I'll save those last few minutes before we discuss as a group about kind of where we think we can go with this work. So for this particular study, we reanalyzed high quality publicly available PKD2 mouse RNA-seq data sets. And so here are the 3 geo entries for the 3 different data sets that I'm going to talk to you about today. All 3 of these are knockout phenotypes. And the first one is a pre-cystic model, and the P70 and the P28 and P21 are both cystic models. And you can see here on the screen available histology images for the P28 and P21. So what we wanted to do here was to try and leverage these data sets to compare differential expression in pre-cystic versus cystic conditions, and then identify and prioritize potential drug targets and drug candidates. I'm not going to spend a lot of time on the differential expression analysis comparisons, because I think what we discovered here is what the field has already known, which is that we found that these differential expression profiles implicated metabolic reprogramming, cyst expansion, and inflammation. So that's of no surprise to this group. However, when we compare across pre-cystic and cystic cohorts, so here you can see a Venn diagram of the 3 different data sets, and I'm showing you the number of significantly differentially expressed genes in each data set compared to its control cohort. And so what you see here is that we see 195 genes that are differentially expressed in the pre-cystic data set compared to the matched control. And we see 22 genes that are differentially expressed that are in common among all 3 of these data sets. So if we restrict to functional enrichment terms and ask questions like pathway analysis and gene ontology, what we find here is that we see enrichment for things like cell cycle, which again isn't all that surprising. But one of the things that we did that is of interest here, going a step further, is that we noticed that in the cystic data sets, we found genes and pathways that were largely reflecting acute kidney injury. And so we saw that in excessive immune, inflammatory, and keratin upregulation. So we wanted to ask the question before we start diving into drug targets and drug repurposing candidates of can we quantify and really nail down that we are in fact seeing an AKI-like signature in those 2 cystic data sets compared to the pre-cystic profile. And so for this, we obtained a list of consistently upregulated and downregulated genes from previous work by Chen et al. And so what they did was they took 4 different high quality AKI data sets. So these were all data sets where there were 24-hour post-injury and time matched controls. And we analyzed those to look for differential expression. And so we and they found 157 upregulated and 88 downregulated genes across all of these different data sets. So then the heat map that you're looking at here on the right side of your screen, what we did was we used these genes to subset the differential expression results that we found in our pre-cystic and cystic data to ask the question of how much overlap there was between the AKI gene set and the PKD gene sets, and to ask the question of if those fold changes of those genes had a more fold change in the cystic compared to the pre-cystic. And so what you can see here in this heat map is each one of the rows is one of those genes that overlapped between the AKI data and one of these pre-cystic or cystic data sets. And each of the columns represents one of the fold changes from one of these 3 groups you see on the screen. So the dark blue column bar is the pre-cystic and the 2 gray are the cystic. And so what we found was that there was in fact more of this AKI phenotype in the cystic compared to the pre-cystic. And so what we decided to do for the rest of our study was really to focus on the pre-cystic transcriptomic signature as our primary signature for drug target and repurposing discovery. And the reason we did that is we thought that this would provide the strongest biological signatures or signals for driving mechanisms. And then using that signal to then filter through our cystic signal. And I'll walk you through that here in just a minute. So we're trying to remove what might be confounded by inflammation or AKI responses. So this pre-cystic data set, this 195 genes here that you see, plus the 38, 22, and 2. That is our signature that we're trying to identify drug targets and repurposing agents against. So before we talk about exactly how I did that, I want to just mention briefly why drug repurposing. And so the reason is because developing new drugs is just really expensive and takes a lot of time. So here's a figure that you can see showing this phenomenon called Eroom's law. So it's the backwards of Moore's law. So this idea that as time marches on, drug discovery gets more and more and more expensive, and we get less and less drugs. So today it costs somewhere, these numbers are still pretty accurate for today, costs somewhere between 12 and 16 years for new drug discovery, and we're looking at billions of dollars. If we look at something that is repurposing or repositioning, so trying to identify new uses for already approved drugs, this has the potential to really cut both our time and cost. So drug repositioning takes somewhere between half to a third of the time and less than a third of the money. So it's a real time saver and a real cost saver. So drug repurposing might be cheaper, faster, and safer. So we have lower cost, faster FDA approval, and reduced risk of failure because these drugs all already have safety profiles. So in the data that I'm going to show you here in a minute, I'm going to show you how we were able to leverage that to, for example, remove drugs that are known to cause liver toxicity, which we know is particularly an issue with PKD patients. So this approach has been used successfully for several other drugs and indications. So one of the ones that's probably the most famous and that y'all would have heard of is aspirin to reduce the risk of colorectal cancer. But there are lots of other less publicized examples. So for example, sirolimus, which is a drug that's used to prevent transplant rejection that's now used for autoimmune lymphoproliferative syndrome, which is a very serious and rare congenital disease. And also Viagra, that's a drug repurposing opportunity that was found through the clinical trials. So thus far, drug repurposing has been very serendipitous. And so our lab and others are developing and leveraging computational methods to try and make this process less serendipitous and to try and prevent adverse events by understanding what these drugs are perturbing in the body and matching accordingly to disease phenotypes. So when we talk about these computational approaches, I want to walk you through a little bit of the framework for the approach that I'm going to tell you about today. And so a lot of the work that we're doing in this study is really based on rich databases of perturbation experiments that have been funded by the NIH over the past decade. And so when I say perturbation experiment, what I'm talking about is a deviation of a system from a certain state that is caused by an outside influence. So essentially here, what you see on the bottom left of your screen is we have a plate of cells, and we've assayed the gene expression profiles of those cells. And then we've exposed those cells to a perturbation. So it could be small molecules, it could be genetic perturbations like a knockdown, knockout, overexpression. It could be antibodies, it could be other environmental stimuli. But then we measure the gene expression again after that perturbation, and we develop a perturbation signature, which you see here in the middle of your screen. And so for each of the genes, then we can identify, did that perturbation lead to an increase in the gene, in that gene's expression, a decrease in that gene's expression, or no change? And that allows us to build these huge databases where we can have tissue or cell type by gene or pathway with respect to a treatment, and ask questions about how those perturbagens actually influence gene expression. So the database that we're working with for this particular study that I'm telling you about today is LINX. And so this is the Library of Integrated Network-Based Cellular Signatures program. And it's a really cool program that folks from 15 different institutions have contributed to. There are 4 different perturbation types, which I mentioned on the previous slide. And there are over 1,000 different cell types that have been perturbed by these different perturbogens. So the folks here assayed different signature types, including transcriptomics, proteomics, binding, epigenomics, and imaging. And they've developed a bunch of different tools to help people analyze that data. So what we did was we took our RNA bulk profile from these public mouse data, so that P70 pre-cystic data set that I told you about, where we had developed that gene signature that we felt like really captured early PKD gene expression changes in this mouse model. And we asked the question using different computer science and mathematical models about how kidney cell lines that were treated with drugs from this LINKS program, how well we thought that those drugs were predicted to flip the PKD signature. So we're looking for drugs that do the opposite. So we want to return those cells and their gene expression profiles to a more normal or quiescent stage. And so when we did that, you get back a lot of different drugs that are possibilities here. And we first restricted that list to those that were already FDA approved. So a lot of the compounds and molecules that are in these screening libraries haven't actually been FDA approved yet. And so we filtered those out. That's something that we could look at more in the future. But for this particular study, we were really laser focused on drugs that already have FDA approval. We also filtered out drugs that we know have black box warnings for severe adverse liver drug events, because this is a problem for PKD treatments and PKD patients. And we also, so we had some drugs that we put on a separate list that we thought might be involved in, might cause some liver phenotype aggravation. Some of our other top candidates were things like calcium channel blockers and phosphodiesterase inhibitors, which we were concerned might further increase cellular cyclic AMP levels and exacerbate PKD-associated cellular phenotypes due to defective PC1 or PC2 calcium sensing and transport. So we also pulled those out into a separate list. So what we were left was several different drugs, which I'm showing you here, our top 25. And this is called an alluvial plot. And so the drug is on the left here. And then these colored lines represent how much of the profile that drug was able to reverse. So the hotter or more yellow the color, the better the job that drug does of reversing the profile, and they flow over here so you can see the different mechanisms of action for which those drugs are FDA approved. And so remember here, this is based on the pre-cystic profile. So what we then wanted to do, oh, I forgot to tell you about the arrows. So the red arrows here and the purple arrows here have to do with drugs that have previously been explored for PKD treatment. So that's the red arrows. There's either a clinical trial or preclinical studies where people have looked at that particular drug and how PKD phenotypes respond to that. And then the purple are if those drugs have been explored for chronic kidney disease or acute kidney injury. So from these drugs, we further filtered and prioritized these from the pre-cystic drug targets and said we also want these drug targets to be active or upregulated in the cystic data sets that we have as well. And so we thought that these might then be drugs that would be effective for both earlier and later in disease, because we've thus far been focused just on the pre-cystic data sets. So this left us with 16 different drugs with 28 unique cystic upregulated drug targets that we predict would target the PKD phenotype in both pre-cystic and cystic disease. So I want to just briefly highlight a couple of these before we wrap this up and kind of talk about what we're doing next. So the first of these is pravastatin. So this is one of our prioritized candidate, and it's really exciting because there's actually 3 different clinical trials that already are using pravastatin for PKD. So there's a phase 4 ADPKD trial, a phase 3 ADPKD trial in children, and a phase 2 trial that's in combination with acidosis treatment. In this case, it's sodium citrate for ADPKD patients with worsening CKD. And so the phase 3 trial has completed, and they showed that the % change in total kidney volume corrected for height was less in the patients on pravastatin. Other studies in animals have been kind of split on its effects on renal function and volume, but there is agreement that it's associated with significantly decreased urinary protein excretion. So we were really excited to see this because it helps us to demonstrate that this process is able to identify drugs that make sense for PKD clinical trials. The second drug that I want to highlight for you is fluvastatin. So this is another statin. And statins in general have been of interest for treating PKD because of the anti-proliferative, anti-inflammatory, and antioxidant properties. So this belongs to that same class of drugs as pravastatin. They, in general, statins, we found papers back to 1995 identifying that statins might be helpful in PKD. But again, another drug in that same class that we identified as being a top prioritized drug. The next drug that I want to highlight is fenofibrate. And so this particular drug is an agonist of PPAR alpha, which promotes fatty acid beta oxidation and oxidative phosphorylation, which are both pathways that have been implicated in ADPKD. And so we think that this makes fenofibrate an attractive candidate for ADPKD. And in fact, there's a study where folks have looked at PKD1 RCRC mice, and they showed that by giving those mice fenofibrate, it reduced renal cyst volume proliferation and infiltration of inflammatory cells, as well as reduced bile duct cysts and liver inflammation and fibrosis. So again, more confirmation that using a completely orthogonal approach, using just the gene expression profiles, that we were able to identify fenofibrate. Paclitaxel is also one of the drugs that's NR16. And so this is one that's really interesting because of the drug targets that it upregulates, including KIF1A and TUB-TUBB, which are both known to be upregulated in different cystic data sets, and is used in cancer. So I want to call your attention to just one more drug before wrapping this up. And this is one that we don't have a direct connection to PKD, but we can speculate as to why it might be showing up in our analyses. And so the reason, so here bromocriptine has been suggested for repurposing in cancer for mechanisms that are known to overlap between cancer and PKD. And so it's been found to have a strong affinity for NF-kappa beta, NF-kappa B, which inhibits expression of pro-inflammatory and proliferative interleukins, tumor necrosis factor, and other factors that have been shown to be upregulated in PKD. So interestingly, there's a study that has tested not in PKD, but in cancer models, paclitaxel and bromocriptine together as an anti-cancer combination therapy. And so that's something that I'll touch on here briefly in just a minute, but it's something we're interested in, is trying to work on strategies for identifying drugs with polypharmacological properties or for use as combination therapies, because in other diseases, this has been shown to reduce side effects and increase efficacy. And so we're interested in trying to work on that in the future. So to wrap up this section of my talk, I just want to say that this study was done by reprocessing and analysis of publicly available data sets. And so these are uses that extended beyond the original analyses that were conducted on these data sets, and really, I think, highlights the importance of public data repositories for increasing knowledge around PKD. We really focused here on that P70 data set because the tissue collection was at the pre-cystic or cyst initiation stage, and we felt that that presented an opportunity to identify early molecular changes associated with cystic kidney disease, and also potentially confounding signatures associated with full cystogenesis. So by being able to compare across these multiple data sets. Out of these top 30 drugs that were already approved for other indications from our signature reversion results, we found drugs with lots of different mechanisms of action. A literature search, we were able to find at least one paper that implicated these mechanisms of action for treatment of PKD in one way, shape, or form. And so we found, our approach identified these drug repurposing candidates and other novel opportunities for follow-up. So I didn't dive into all of the drugs today, but we have a whole list of other drugs that would be potential candidates for future studies. So we think that additional drug candidates might be identified by investigating more targets, polypharmacological properties, combination therapies, or cross-disease relationships, and that efforts to refine these candidates by prioritizing attributes associated with clinical success and model selection and interpretation are really important. And so I touched on these throughout the talk, but I just want to kind of use my last 5 minutes to talk about these a little bit more, because I think this is a place where there are opportunities for synergy and collaboration, and it's also kind of where my research group is hoping to go over the next few years. So I think the obvious thing here is we really want to perform drug target and repurposing analyses in other PKD models. I showed you in 3 different PKD2 data sets. We'd love to perform these same analyses in PKD1 or in AR-PKD models. 2, incorporating cross-species and model considerations. So we've cured disease in mice many, many times. So the question here is, how can we stack the deck in our favor to identify drug targets that we think are going to work in both mice, which is going to be really important for those preclinical studies, or in other model organisms, as well as in humans. So, and how can we reduce risk, time, and money to prioritize targets that are conserved? So just really briefly, I will say that we haven't applied this yet to PKD, but we've applied approaches in, for example, in muscle, to ask the question of, can we identify exercise patterns in humans, which you see in this top left box, where each of these purple-green pairs represents pre and post gene expression patterns that we identified in humans. And here in the top right box, you're looking at this figure that shows you what those patterns are. So what is the gene expression analysis? If we look at the genes, what do the Go terms look like? And we find that these patterns capture biological processes we'd expect to see associated with exercise. And so we can actually take that particular one of those patterns that we've identified as being associated with pre or post exercise in humans, and we can computationally project it onto a mouse, and we can try and figure out which parts of a mouse profile that are changing are similar to that of a human. So some of these other patterns that don't show exercise change might be due to batch effects from the sequencing. They might be due to age. They might be due to other conditions that are not specifically associated with the actual exercise pattern. And so what we can do here is then take those patterns and identify which are consistent across species. And so this approach has advantages because, unlike one-to-one gene equivalences, here we can capture variation across data sets, even if it's not the exact same genes acting in the same way. We're also working on other tools to develop and facilitate cross-species analyses. And so here's one that we released out into the wild just last week. It's an R bioconductor package with a Shiny app to come. We're doing some more testing and adding some more instructions for the Shiny app, but the R package is available, and it facilitates cross-species analysis for transcriptomics. And so we have folks in our lab who are continuing working on this, and we're going to add functionality to be able to look at targets and regulators of genes across species, as well as gene regulatory networks between species and tissues. And so we have funding to do that. And just as one example, I just pulled right before the talk. If you look here at across transcriptomics data available in BG, which is this huge cross-species transcriptomics repository, I just asked the question of, in mice, what does PKD1 and PKD2 expression look like? And so you can see here in green is kidney, purple is brain, red is heart, and yellow is liver. So different tissues that would be of interest to the PKD community. And you can see, for example, that depending on which mouse you're working with, the expression in the brain or expression in the kidney is more heterogeneous in this particular database. So just as a quick proof of concept. So I mentioned that we're interested in trying to identify combination therapies. So we're working on machine learning and multimodal network approaches. And I have some like backup slides we can talk about as a group if that's of interest. And also thinking about cell type and tissue specific gene regulatory states. So I mentioned at the top of the hour about how PKD1 and PKD2 are widely expressed, as are the proteins. So we think it's really critical to try and understand that expression, because we can learn something about why some cells that do have changes in PKD 1 or 2, why are those cells not showing a phenotype or a molecular phenotype? And we can also learn something about PKD patient risk for other diseases. The other area that we're working on is really thinking about biological sex and how it's associated with PKD. So while males and females are equally affected, being male is a risk factor for progression. So men demonstrate faster loss of renal function and earlier onset compared to women. This has been recapitulated many times in mouse models. Here's an example in a PKD1 model showing that onset and severity are sooner and more rapid for male mice compared to female mice. So not only is this important for trying to understand the mechanisms of disease, but also sex-based adverse events are a leading cause of drug recall. So in fact, in many years, it's the leading cause of why drugs are recalled is because of sex-based adverse events. So we've written a review about this. If you're interested, a little shameless plug, you can go check it out. But we're really interested in trying to apply that and understand what's happening here in PKD. And so finally, this is my last real slide, is thinking about how to put all of this together. So we're envisioning putting together different models where we're able to look at drugs, drug targets, and drug repurposing candidates and ask questions about which of those are able to reverse or hit targets that are important for PKD, but then also refining them based on their known or predicted toxicity or side effects, or their ability to perturb or not perturb things like sex bias gene expression. And I've got some really cool preliminary data from a grad student in my lab about some of this that I'd be happy to talk about in the discussion if there's interest. So I think I will just buzz through this in the interest of time and say genomics and data science are awesome. And I think that there's a lot of potential here to apply them to PKD. And then just a shout out to my collaborators and the folks in the lab who are in yellow are the ones who did the heavy lifting on this, but particularly Lizzie, who's the second one here next to me on the left on the slide. So thank you so much for your attention, and I'm looking forward to discussing further with you.
Hey, Brittany, thank you very much. So if for questions, please raise your hand, and encourage you to ask your question, unmute yourself and ask your question. I'm going to start off with a lot of the drugs that you showed there all seem to have errors that pointed to that group of genes at the bottom right. So have you looked, and I guess this is part of your prediction model, right? So your goal would be to reverse that whole series of 16 genes, or how many ever it was. Have you looked at what might be the effect of combinatory drugs on these genes?
Yeah, so we want to do that. So in fact, we've started to do that a little bit. Let me see if I can pull up. I have a slide.
I know you didn't go through all 90 of these, but yes, okay.
Yeah, I have like 90. So let's see. Let me see if I can. Oh, I lost the. I'm so sorry. I lost my share. There we go. Okay, so can you see it or no?
Yeah, we see it.
Okay, so one of the things that we've been thinking a lot about here is with respect to combinatorial. And so there's a lot of different ways one could do this. This is actually a figure from a grant proposal that we put in a couple months ago. And really this idea of if we combine drugs, so here are some of the drugs that I showed you today, not all of them, but just some of them here on the left side and on the right side. And so where they intersect is our prediction of whether or not those genes have orthogonality. So meaning that they are very different from one another and perturb very different genes, and whether they have discordance. So meaning that those drugs one drug would reverse part of a profile and the other drug would reverse a different part of the profile. And so what we see here is, for example, paclitaxel and crizotinib or fluoxetine here, the big purple circles to the right are the ones that we've predicted were going to be the most different from each other and reverse the most of the profile together. There are other ways one can do this, and we're working on this and other projects in the lab and would love to apply it here, where we're working through multimodal network approaches to hit the same targets or same pathways through drugs. So there's some mixed feelings in the field about whether or not it's better to reverse more different of the profile or hit the same parts of the profile, but with lower doses of drugs. And that has to really do with both efficacy and toxicity.
Gotcha. Dr. Khan, unmute yourself, please.
My question is about the statins, and a lot of patients are on statins. Is there a database of statins and PKD patients?
Yeah,
And
that's a great question. So that's something that Michael Murug, who I think I saw on the call, and I have talked about is if we can access some of the different databases through the CRISP study or HALT or other databases to ask questions about that. It's not something we've done yet, but I would like to do that. And we've talked about that for some of the drugs that are expected to be, being taken by a larger percentage of the population. Yeah, we haven't done it yet, but I would like to.
the part two of that was are there differences in the statins?
Yeah.
Were you because some patients can tolerate some and some they can't. Is there difference? Is there real differences there? And may they have a different phenotype?
That's a great question. So some of the statins, the statins do have different ranks on our list, and they're predicted to reverse different common drug targets, but then some of them are also known to hit different drug targets, or that we can show computationally in these databases do in fact reverse different parts of the profile. So that's something that we've thought about and would like to dig into more. As you could imagine, a study where you take all of the statins for which we have data and ask questions about the known side effects of those and the tolerability of those with respect to like the percentage of the profile that's able to be reversed. So it's a good question.
No questions?
Christian Hanna from Mayo Clinic, pediatric nephrology. Thank you for the talk. Just to clarify regarding the phase 3 trial of pravastatin, I'm not aware about any current study in pediatric ADPKD. Can you clarify? I know that there's a study that done at Colorado Children's in the 2014 publication, but if there's any ongoing trial at the moment?
I'm not aware of an ongoing study. The one that I was talking about is the one that you're talking about that already completed. And so they hit several of their indicators and did show that there was a statistically significant difference in the pravastatin versus the placebo group. But I'm not aware of any current. The other two trials that I'm aware of are at the adult level.
Thank you.
Dr. Wellen.
Thank you for just a spectacular talk, really exciting. I wonder if you could predict the future a little bit for us. When I think we've just scratched the surface in genomics and different models of PKD. You went after PKD2 and what you called early onset. I'm sure you'll get some quibbles about that. But as we see different models come together, how do you predict we're going to put all these models together when they may reflect different stages of the disease or what people call different stages of the disease? What's next in this putting all the data sets together?
Yeah, that's a great question. So I guess I have a couple of answers or at least, thought experiments to put back to the room. So so I think that it really underscores the need to have more data from humans. So I don't know if he's on this call, but the Humphrey Lab, of course, published a paper not too long ago where they looked at single nuclei RNA-seq profiles. Now those were at late stage PKD. Myself and Dr. Murag on the call would really like to do single cell sequencing from urinary samples from patients. And so that's been done and published not in PKD, but in AKI, in patients with COVID, in healthy controls, and in patients with CKD, where you can successfully get cells from patient urine and then do single cell sequencing, and you can computationally then or bioinformatically take out, for example, cells that come from the bladder, and you can identify cells based on their expression profiles that are from the kidney. So I think that what we really need are more data sets in that vein in the human world, so that we can place these different animal models in context. The other answer that I would give you is that that's where I think some of these different approaches that we and others are trying to do to really try and understand, so which parts, if you will, of a model profile are most relevant for the human disease are going to be really important. And so again, it calls back to we need more human data. But I think that by comparing these different profiles, we can start to get a better look at what molecular phenotypes are conserved across different species. And maybe you were one of the reviewers of our manuscript. They asked us why we didn't use cat or pig. So which there is pig data available that just came out that we just found. So we're working on that. So
Dr. Prune.
Hi, I have a question which might be a slight variant on Paul's. So how will you distinguish initiating events versus subsequent downstream programs of inflammation, fibrosis, apoptosis, all of that stuff that kicks in?
Yeah, I think that's a great question. So, this particular project was kind of our first attempt at that, where we used what was classified as pre-cystic based on histology at the time of resection. I think this calls for more data at more time points in order to be able to do that. So, here we had one, pre-cystic time point where we were able to then cross-reference with gene sets that we know are common to acute kidney injury and inflammation and processes like that. And of course, this is not going to be a perfect analysis, and without additional data sets, there's only so much we can do. But I think that additional data would help us to further refine, and that we can also try and ask those sorts of questions with some of the fibrosis. So yeah, I think I would like more data earlier in order to do that. Brad's laughing because I'm always like, let's RNA-seq it. That's kind of what I'm known for. But I'm sincere when I say that, because I think if we have more data earlier on more models, then we can start to tease that apart in collaboration with histology and with other other molecular experiments.
Questions? Not hearing any. Brittany, we normally at the end of this reverse the question, and that is basically, what can the center do to help promote or advance your research program other than sequencing
Everything?
Yeah. Which, maybe that's something we need to consider as a PKD RRC, whether, generating data sets from PKD patients might be something that we need to tackle in the future, although that's a challenging thing to do.
Yeah, I definitely am not sitting here, glib about the challenges of that. Working with Michael, I definitely have had a front row seat in, what the challenges are there. I think, for me, I think there's some Certainly, yes, I want to sequence everything, all the models at different time points, and I'm interested in trying to understand. I think the sex bias is something that I'm really excited about, and trying to understand differences in the presentation of the disease and progression of the disease. And so a lot of the data sets, you know, we've been going through and kind of dumpster diving through the literature and through Geo to try and find data. A lot of the data sets, you know, only have one sex. So I guess I would, you know, politely suggest thinking about that in future experiments. You know, maybe efforts to try and curate some of the data that's available already to make it that less of a lift for other people. So like this is what we do, it's kind of our thing, but I think that most labs now are interested in trying to incorporate gene expression into their studies, or ATAC-seq or whatever, you know, pick whatever your favorite omics assay is. And so some of these are challenging to find, and challenging to get the metadata, the information about the samples and about time points and things like that. And so it would be really cool to have, you know, a database or, you know, more curated resources there. That would be awesome. I guess those are my three things: human samples, female samples, and database or more information about data sets.
All right, Brittany, I want to thank you for taking the time to meet with us and the audience for a robust discussion. I appreciate you doing this.
Thank you so much.